I would like to thank the organizers for allowing us to introduce VRON 0200. This is a therapeutic HPV vaccine with an intrinsic checkpoint inhibitor that elicits a very potent CD8 T-cell response, which achieves sustained declines in viral loads in a preclinical animal model. Let me start by declaring conflict of interest. 3 of the authors of this abstract have an interest in Virion Therapeutics, the owner of this vaccine. CD8 T cells become exhausted during a chronic HPV infection, and that results in loss of viral control. Immune modulators and vaccines have been tried to treat chronic HPV infections, but they have shown limited clinical benefit. For example, T cells cannot be rescued with a PD-1 checkpoint inhibitor, for during exhaustion, T cells differentiate in a way that they undergo epigenetic changes. And once they reach this point, nothing will revert them towards functional T cells. In addition, PD-1 blockade has off-target effects, which are too risky in a healthy population. So the goal of our vaccine has been to stimulate T cells that are still circulating and that are naive. The infection will trigger mainly activation of T cells to immunodominant epitopes. And over time, they'll become exhausted. But some of the naive T cells to subdominant epitopes are still present. And we aim to stimulate them under the assumption that they will be fully functional and thereby able to control viral infection. Our vaccine is composed of 3 parts. The vaccine antigen is a consensus sequence of coin polymerase for genotypes A through D. We purposely did not use S antigen, for it's present at very high levels in chronically infected patients. So chances are that all T cells with this antigen are fully activated and exhausted. We optimized the sequence for binding to multiple HLA genotypes using prediction algorithms. And it turned out that this included regions that are already known to prevent flares and viral breakthroughs. As vaccine carriers, we use chimpanzee-derived adenoviral vectors to which humans do not have pre-existing neutralizing antibodies that could dampen vaccine immunogenicity. And we developed 2 serologically distinct vectors for optimized prime boosting. The adjuvant is herpes simplex virus glycoprotein, also called GD. It is genetically encoded by the vaccine, so it is only present locally at the site of injection and in draining lymph nodes, so that off-target side effects are highly unlikely. In addition, we developed a challenge model based on an AAV8 vector expressing the overlength HPV genome. Upon intravenous injection into mice, this AAV8 vector induces a production of HPV particles that can be detected in serum. During T cell activation, antigen-presenting cells will present a peptide from the antigen on the surface of an MHC class 1 molecule to T cells, which this particular complex binds to the T cell receptor, which together with co-stimulators will then signal activation of the T cell. This is regulated by a number of other molecules. In addition to the co-stimulators such as CD28, we have co-inhibitors, for example, BTLA, the BAT lymphocyte attenuator. BTLA binds to the herpes virus entry mediator, which is expressed on antigen-presenting cells. And this particular molecule can also bind light, which is a co-stimulator. And the 2 molecules bind to different sites on HVM. So you can have a trimolar complex in which inhibition through BTLA prevails. BTLA, although the exact mechanism isn't known yet, is assumed to dampen T cell receptor signaling downstream from the T cell receptor. As you can see in the middle, herpes simplex virus GD is structurally similar to BTLA, and it can bind to HVM to the BTLA binding site. It has higher affinity to HVM than BTLA has, so it can outcompete BTLA, so that then inhibition through BTLA no longer takes place, and instead you get activation through light. So in our vectors, we encode GD, into which we fuse the antigen away from the HVM binding site. Once the protein is produced, some of it is misfolded, it gets degraded into peptides. Some of the peptides associate with MHC class 1, which then go to the cell surface for recognition by the T cell receptors. We also get full-length protein that can be transported to the cell surface, where it binds to HVM, preventing binding of BTLA. So we now get signaling through the T cell receptor, which is not blunted by signaling through BTLA. For the initial proof of principle studies, we made 3 vaccines, which we, based on 3 different sequences of HPV, we used polymerase. It's a very large protein, so we broke it up into an N terminus and a C terminus, which were both cloned into GD. We also made a core vaccine. We immunized mice with these vectors, and then we tested T cell responses in blood. And as you can see here, we got a very strong response to the GD pollen vaccine, over 7% on average. We also got a good response to Pol C and lower, but still readily detectable responses to core. We then looked into the liver using a tetramer that recognized an epitope within the Pol N part of the vaccine. Mice were either injected with an AAV8 vector at 10 to the 10 genome copies or 10 to the 11 genome copies. These animals developed high loads of HPV particles in blood. They were then vaccinated 4 weeks after injection of the AAV 1.3 HPV vectors. And other mice were not injected with AAV, but just with the vaccine. And then we measured T cell responses in liver several weeks later. And as you can see, we get very good responses in liver after vaccination. On average, 50% of all T cells in liver are directed to this one epitope within Pol N. And even in presence of high loads of HPV particles, we still get very robust T cell responses to HPV in liver. Most importantly, when we inject mice with the AAV particles and then vaccinate them 4 weeks later, we see a steady decline of viral loads in serum in a lot of the animals. So what I showed you so far were results with these vaccines. We didn't want to go ahead with 3 vaccines, so we combined the most immunogenic sequences of Pol N with some sequences from Pol C and core into one vector, which we called GD-HBV2, which is VRON 0200. In addition to the GD version, we made a version without GD to confirm that GD was essential for the efficacy of vaccines. And we made a vaccine from which we eliminated some of the highly dominant, immunodominant epitopes of Pol N, which we called HPV SD, and SD stands for subdominals. And then we analyzed the T cell response to these vaccines in blood, spleen, and liver by intracellular cytokine staining for interferon gamma. We did some MHC tetramer staining experiments, and we mapped the epitope for intercellular cytokine staining to determine which parts of the vaccine insert was being recognized. In addition, we conducted the challenge experiment in which mice were injected with the AAV8 1.3 HPV vector at 1 × 10⁹ viral genome copies intravenously. And then 4 weeks later, they received a single dose of 10 to the 10 virus particles of the adenoviral vectors. This shows you the epitope recognition profile of the T cell response to these 3 different vaccines. Pol N, as you may recall, only contains this part of the protein, and that's where we see the responses. The GD-HBV2 vaccine contains core Pol C and Pol N, and as you can see, we see responses scattered throughout the protein. The subdominant epitope vaccine also has most of these segments. It lacks this particular region, which has very immunodominant epitopes. And again, we see robust responses to various epitopes. We want to make sure that the new vaccine GD-HBV2 was similar in immunogenicity to GD Pol N, with which we achieved a very good antiviral response. So we conducted prime and prime boost studies. This is the result after prime, and you can see both vaccines give us similar T cell responses. The GD-HBV SD response is much weaker. After the boost, we see a slight increase in responses, which are best in some of the animals that got the less immunogenic vaccine. This was confirmed with tetramer staining for GD Pol N and GD-HBV2. Both gave approximately the same frequencies of T cells in blood as well as in liver. Most importantly, when we injected mice with the AAV8 1.3 HPV vector, we saw within 4 weeks strong high titers of viral loads measured as DNA copies in serum in all of the animals. We then vaccinated the animals, and we saw bio-naive mice showed a slight increase in titers over the following 4 weeks, which then stabilized a fairly robust decrease in viral loads upon immunization with the GD pollen vaccine and the GD HPV2 vaccine. GD was clearly important, for if we only used HPV2 without GD in our vaccine, we saw a decline, but it was much less robust. And also, we saw a decline with the vaccine from which we had eliminated the dominant epitopes, but here the decline was delayed. We measured in the same mice the CD8 T cell responses. They were very potent upon vaccination with GD pollen or GD HPV2, less robust upon vaccination with the HPV2 vaccine without GD, and fairly modest upon vaccination with the subdominant epitope vaccine. CD4 responses were negligible. What was important is that CD8 T cell responses in blood strongly correlated, inversely correlated with viral loads. And this is shown here, indicating that CD8 T cells contributed to a reduction in viral loads in serum. So in summary, the vaccine elicits potent and broad CD8 T cell responses to HPV core and polymerase. The vaccine induced CD8 T cells traffic to the liver. We can achieve a multi-log decline in HPV DNA viral loads after a single IM injection. And in order to get an optimal antiviral activity, GD is required. And the antiviral load declines are inversely correlated. So the antiviral loads are correlated with vaccine-induced CD8 T cells, indicating that these cells contribute to reduction in viral loads. At this stage, we are planning on a phase 1 trial in HBe antigen negative patients on nucleoside therapy, and we are planning on testing both prime as well as prime-boost regimens. I would like to thank 3 additional people from my lab who contributed to these studies. Thank you for your attention.